Have you ever wondered what makes living organisms different from non-living things? The answer lies in cells, the basic units of life. While some organisms, like bacteria, consist of just a single cell, unicellular, others, like humans, are made up of trillions of cells working together, multicellular. Even a single cell is capable of performing all essential life functions. That's why the cell is considered the fundamental unit of life. The first person to observe a living cell was Anton von Leeuwenhoek, while Robert Brown later discovered the nucleus. Thanks to advancements in microscopes, especially the electron microscope, scientists have been able to study cells in much greater detail. In 1838, Matthias Schleiden discovered that plants are made of cells, and a year later, Theodor Schwann found that animal cells also have a defined structure. He also identified the plasma membrane and the cell wall in plant cells. Their combined research led to the formation of the cell theory, which states that all living organisms are made of cells. But there was one big question left unanswered. How do new cells form? In 1855, Rudolf Virchow provided the missing piece of the puzzle. He proposed that new cells arise from pre-existing cells, summarizing it in the famous phrase, omnis cellula e cellula, which means every cell comes from another cell. Today, the modern cell theory states, all living organisms are made of cells and their products. All cells arise from pre-existing cells. Now let's compare plant and animal cells. If you look at an onion peel under a microscope, you'll see plant cells with a rigid cell wall surrounding the cell membrane. But if you examine human cheek cells, you'll notice that they lack a cell wall and have only a cell membrane as their outer covering. Despite these differences, both plant and animal cells have a nucleus, which contains DNA, the blueprint of life. Cells can be classified into two major types. One, eukaryotic cells. These have a membrane-bound nucleus and organelles like the mitochondria, endoplasmic reticulum, and Golgi complex. Examples include plant and animal cells. Two, prokaryotic cells. These lack a well-defined nucleus and membrane-bound organelles. Examples include bacteria and bacteria. Now let's talk about cell size and shape. Did you know that the smallest cells belong to mycoplasma, measuring just 0.3 micrometers? On the other hand, the largest single cell is the ostrich egg. Human red blood cells are about 7 micrometers in diameter, while nerve cells can be extremely long, extending over a meter in some cases. The shape of a cell depends on its function. It can be disc-shaped, polygonal, columnar, cuboidal, or even irregular. Now let's focus on prokaryotic cells, which include bacteria, blue-green algae, mycoplasma, and P. yellowstralo. These cells are typically smaller and multiply rapidly. Bacteria come in different shapes. Bacillus, rod-shaped, coccus, spherical, vibrio, comma-shaped, spirillum, spiral-shaped. Although prokaryotic cells differ in shape and function, their basic structure remains the same. Most of them have a cell wall surrounding their plasma membrane, except for mycoplasma, which lacks a cell wall entirely. Inside the cell, the cytoplasm contains genetic material in the form of circular DNA, but unlike eukaryotic cells, it's not enclosed by a nucleus. Some bacteria even have extracircular DNA, called plasmids, which can give them special traits like antibiotic resistance. Prokaryotic cells also have a unique outer envelope with 3 layers. Glycocalyx, the outermost layer, which can be a loose slime layer or a thick capsule for protection. Cell wall provides structural support and shape to the bacterium. Plasma membrane, a selectively permeable barrier that regulates the exchange of substances. Bacteria are further classified into gram-positive and gram-negative based on how their cell walls react to gram staining. Gram-positive bacteria have a thick peptidoglycan layer, while gram-negative bacteria have a thin peptidoglycan layer with an additional outer membrane. Now let's talk about some specialized structures in prokaryotes. Mesosomes. These are folded extensions of the plasma membrane that help in DNA replication, respiration, and cell wall formation. Chromatophores, found in cyanobacteria, these contain pigments for photosynthesis. Ribosomes, these non-membrane bound organelles are involved in protein synthesis. In prokaryotes, they are made of 50S and 30S subunits, forming 70S ribosomes. Prokaryotes also have storage structures called inclusion bodies, which contain reserve materials like glycogen, phosphate granules, and cyanophycin granules. Some bacteria even have gas vacuoles, which help them float in water. For movement, many bacteria use flagella, thin whip-like structures made of 3 parts: filament, hook, and basal body. The number and arrangement of flagella vary across different bacterial species. Eukaryotic cells are the foundation of life for protists, plants, animals, and fungi. What makes them special? Unlike prokaryotes, they have membrane-bound organelles, a well-defined nucleus, and a complex cytoskeleton that gives them structure and support. Their genetic material is neatly organized into chromosomes within the nucleus. But did you know that plant and animal cells aren't identical? While plant cells have cell walls, plastids, and a large central vacuole, animal cells have centrioles, which are missing in most plant cells. Now let's dive into the cell membrane, a dynamic, flexible boundary that controls what enters and exits the cell. It's made up of lipids and proteins, with phospholipids arranged in a bilayer, polar heads facing outward and hydrophobic tails tucked inside. Proteins in the membrane play key roles. Integral proteins are embedded, while peripheral proteins sit on the surface. The fluid mosaic model, proposed by Singer and Nicholson in 1972, explains how the membrane's fluidity allows proteins to move laterally. And, of course, the membrane is selectively permeable, allowing passive transport like diffusion and osmosis, or active transport that requires energy, ATP, to move molecules against their concentration gradient. Moving beyond the membrane, we find the cell wall, a tough, non-living structure found in plants and fungi. It gives shape, protection, and allows cells to interact. Different organisms have different cell wall compositions. Algae have cellulose, galactans, and calcium carbonate, while plant cell walls contain cellulose, hemicellulose, pectins, and proteins. As a plant cell matures, the primary wall strengthens into a secondary wall. The middle lamella, made of calcium pectate, acts like glue, holding neighboring cells together, while plasmodesmata connect their cytoplasm for communication. Next, we have the endomembrane system, which includes the endoplasmic reticulum, ER, Golgi apparatus, lysosomes, and vacuoles, all working in sync. However, mitochondria, chloroplasts, and peroxisomes are not part of this system. The endoplasmic reticulum, ER, is like a cellular highway, a network of tubules dividing the cell into luminal and extraluminal compartments. The rough ER, RER, is studded with ribosomes and specializes in protein synthesis. In contrast, the smooth ER lacks ribosomes and focuses on lipid synthesis, including steroid hormones in animal cells. The Golgi apparatus acts like the cell's post office, packaging and modifying proteins for intracellular use or secretion. Vesicles from the ER fuse with the cis face of the Golgi, where proteins undergo modifications before being sent out from the trans face. The Golgi is also responsible for making glycoproteins and glycolipids. Now let's talk about the cell's recycling center, the lysosomes. These membrane-bound vesicles originate from the Golgi and contain hydrolytic enzymes like lipases, proteases, and carbohydrases that function in acidic conditions. They digest carbohydrates, proteins, lipids, and nucleic acids, basically breaking down waste and worn-out cell components. Moving on to vacuoles, these storage units hold water, sap, and waste. In plant cells, vacuoles can occupy up to 90% of the cell's volume. They are enclosed by a membrane called the tonoplast, which regulates ion transport. In amoeba, a contractile vacuole helps maintain water balance and aids in excretion, while food vacuoles digest engulfed particles. Now let's step inside the powerhouse of the cell, the mitochondria. These double membrane-bound organelles vary in size and shape, but are usually cylindrical or sausage-shaped. The outer membrane forms a protective boundary, while the inner membrane has enfoldings called cristae, increasing surface area for ATP production, the energy currency of the cell. Inside, the matrix contains enzymes, circular DNA, RNA, and ribosomes, allowing mitochondria to self-replicate through fission. That's why they're known as the powerhouses of the cell. Next up, Plastids, which are exclusive to plant cells and euglenoids. They contain pigments and are classified into three types. Chloroplasts, essential for photosynthesis, containing chlorophyll and carotenoids. Chromoplasts, rich in carotenoids, giving plants yellow, orange, or red colors. Leucoplasts, colorless plastids that store nutrients like starch, amyloplasts, fats, oleoplasts, and proteins, oleuroplasts. Inside chloroplasts, stacks of thylakoids form grana, where photosynthesis occurs, while the stroma houses enzymes, DNA, and ribosomes. Now let's talk about ribosomes, first observed by George Palade in 1953. These tiny granular structures are made of RNA and proteins but lack a membrane. They come in two types, 80S in eukaryotic cells and 70S in prokaryotic cells. Ribosomes consist of two subunits. For 80S ribosomes, it's 60S and 40S, while for 70S ribosomes, it's 50S and 30S. These are responsible for protein synthesis, one of the most vital cellular processes. Supporting the entire cell is the cytoskeleton, a network of microtubules, microfilaments, and intermediate filaments. This framework provides shape, mechanical support, and motility. Cilia and flagella are hair-like structures extending from the cell. Cilia are short and move rhythmically, while flagella are longer and responsible for movement. Both share a 9+2 arrangement of microtubules supported by radial spokes and linkers. The centrosome plays a key role in cell division. It contains two centrioles, which are cylindrical structures made of 9 triplet fibrils of tubulin protein surrounded by pericentriolar material. These centrioles form spindle fibers and the basal bodies of cilia and flagella. Now, let's look at the command center of the cell, the nucleus, discovered by Robert Brown in 1831. This membrane-bound organelle contains chromatin, the nuclear matrix, and nucleoli. The nuclear envelope, a double membrane, separates the nucleus from the cytoplasm. Tiny nuclear pores control the exchange of RNA and proteins between the nucleus and cytoplasm. Inside, chromatin is made of DNA and proteins, histones and non-histones, forming chromosomes during cell division. The nucleolus, a dense, non-membrane-bound structure, is responsible for ribosomal RNA synthesis and is larger in cells with high protein production. Finally, let's talk about chromosomes. They contain centromeres, which hold two chromatids together. Based on centromere position, they are classified as metacentric, submetacentric, acrocentric, or telocentric. Some also have secondary constrictions, known as satellites. And that's a deep dive into the eukaryotic cell, a tiny world packed with organization, efficiency, and life-sustaining processes. Keep exploring biology and stay curious.